properties based on API properties? Thank you.
Thank you. Thanks to the organizers for giving me the opportunity to speak today, and thank you all for indulging me for the next 35 minutes while I talk about selection of oral modified release technologies based on API properties. All right, so just to kind of kick things off, what are modified release formulations? So modified release formulations are dosage forms that have release characteristics of time course and or location that are chosen to accomplish therapeutic or convenience objectives which you can't get with a traditional IR dosage form. These are fairly common. It's about a third of the top 200 prescribed drugs in the United States are classified as modified release, and about a quarter of all oral drugs approved by the FDA over the last, let's call that 15 years or so, were modified release. And these are more complex in that they're engineered to release these drugs in a controlled manner to generate a certain PK pattern to meet clinical needs. So there's generally kind of three types. So conventional delayed release, you have a release profile that looks like this. You have nothing, 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 then ideally it kind of just shoots up, and you end up with a PK profile that looks kind of like that. This one's a bit more complicated where it's delayed and pulsatile, so you have an immediate release profile, kind of nothing, 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 and then the delayed release, and you get this sort of two-humped pattern. And then extended release, where you just release it kind of low and slow, and you get a PK pattern that looks like that. So those are the two general types, and that's the regulatory classification as well. It's either extended release or delayed release. So there's drivers and risks for this. So for extended release, it's usually a reduction in dosage frequency to improve patient benefits. Also, by that, you'll often avoid Cmax-related adverse events. And you can better control your concentration, especially if you have a narrow therapeutic index. For delayed release, you're usually looking to deliver to or avoid a specific region in the gastrointestinal tract. One thing that's actually been kind of more commonly seen more recently is chronotherapy, where the idea is for like certain diseases, you can take the drug at night, you kind of sleep, it releases it at, let's say, 4 in the morning, so you have full therapeutic benefit first thing in the morning. So that's beneficial for like arthritis and some other similar indications. And then for a lot of drugs, you may see some degradation in certain regions in the GI tract. The stomach tends to be very common within its acidic environment, so you can kind of avoid there. However, it does come with some trade-offs. One is it's more complex development compared to an immediate release. You often require specialized equipment and or excipients. Often it takes a couple of shots in the clinic to get it right. I'm not talking about it today, but the preclinical models, are not ideal for modified release development, and the regulatory requirements tend to be fairly conservative, so it kind of puts you in a somewhat of a tough position if you need to move quickly. The next part is, in this case, my apologies, physiological factors. The release rate can really be altered by differences in your food intake, your gastric transit time, your pH, which has a lot of intra and intra-subject variability. And lastly, your tablets can't be crushed or chewed or split, which is certainly an issue with some patients. And then the last part is assuming you're starting with an IR dosage form to just get things moving in the clinic, you really have to understand your PK/PD if you're going to try to bridge from, like, let's say a BID IR to a QD MR. You really have to understand that relationship. Otherwise, you're going to develop something that you hit your targets perfectly, but you got the wrong targets from your clinical colleagues, which isn't good. All right, so what dictates the profile of the modified release formulation? And I think there's kind of two general parts here. One is the release rate, which is controlled by your formulation. Your release location, again, controlled by your formulation. This can be dictated by your design of your delayed release or the duration of release for your extended release. The next part is your regional bioavailability in the intestine, which is a compound property. I'll talk about this later, but this can have a pretty significant impact on your pharmacokinetic profile. One of the key risks that I'll talk about later is, loss of absorption in the colon, and it can also be impacted by your physiological conditions, whether your intestinal surface area, your water volumes, transporters, transit times, what you've had to eat that morning. And also, for some drugs, you'll actually see higher bioavailability. Actually, the opposite profile, we'll see higher bioavailability in the lower GI simply because you're having some regional intestinal degradation in the upper GI. The last part is sort of your systemic pharmacokinetic parameters, which is again compound dependent, but will also have some impact by your formulation because your relative rates of input and elimination will somewhat dictate that profile. All right, so first things first to understand for modified release formulation is understanding your API properties and making sure you're actually, before you even start, picking a good candidate. So here are this is a paper some of our colleagues at Pfizer put out in 2005 looking at some of the physiochemical properties. So the first, and with kind of relative risks. So the first risk highlighted here is the dose. If you have a very low dose, you have a lot of complexity. It's just simple, what I would call low dose drug development. If you have a moderate dose, you probably are in a decent spot. However, if you have a higher dose, say greater than 250 mg or above, you may run into some issues with modified release development because you're going to need a high drug load tablet. You're not going to have a lot of room for some of your excipients, and actually, what I'll show later is that actually will remove some of your options as well from a formulation perspective. Next is kind of your dose to solubility ratio. If you have what I would say a highly soluble to pretty soluble drug, you're probably in a decent spot. Your drug is going to get you would expect your drug to be solubilized, and you have a number of technology options available. As you start getting into more insoluble drugs, the challenge kind of comes in because not only are you going to think about modified release, you also need to kind of layer on the fact that you may need drug solubility enhancement as well. And the last part is just kind of, stability. If you're seeing kind of a drug that's generally stable, happy with most excipients, you're in a good spot, but at the same time, you could be limiting yourself there if you start seeing drug to excipient interactions. And then, so once you kind of go from there, then you have to start thinking about your biopharmaceutics properties. And I'll talk about a few things here. First is sort of your human half-life. And this is sort of assuming more an extended release drug, but targeting once daily. Delayed release, some of these properties can be a little different. First is sort of your half-life. My apologies. First is your half-life. Ideally, you'd like kind of a moderate half-life for once daily. If you have kind of a longer half-life, you probably aren't going to need modified release or certainly extended release. Certainly a workable range is 2 to 3 hours. However, it could become challenging if it's less than that if you're going for once daily administration simply because, the drug's just going to get eliminated that quickly so generating your desired plasma concentrations could be just very challenging if it's being eliminated as fast or faster than the drug is being kind of absorbed in. The next part is your colonic absorption. Workable range is 30% or above. Ideally, you'd like it to be 50%, if not higher. If you're less than that, you're probably going to be in a challenging spot for an extended release because you're just going to have a complete drop-off of absorption at that range. So you're either going to have to dose really high, and there's been a number of examples in the literature of compounds where they've tried multiple iterations, but they've never actually gotten an extended release to work. From a metabolism point of view, you want to be kind of in a good spot there, minimal first pass, hopefully linear PK. If not, things get a little tricky. If you're seeing a lot of high metabolism degradation, PGP efflux, again, this is going to be a challenging compound to develop. in general, and modified release will make it even harder. Bioavailability, standard kind of considerations here, you'd like a moderate to high bioavailability for your compound. For ideally, if you can kind of make something moderate work, and again, a compound with low bioavailability, you're in a tough spot to begin with. And then the last is sort of building an IV-IVC. I don't really touch much on this, but basically, ideally you would like to build some sort of correlation between your in vitro data and your in vivo response in humans. If you are seeing that, it makes your life a lot easier from both a formulation development perspective, and as you move into late phase, it gets a lot easier. And I'm being a little flippant here, if you don't have an IV-IVC, the second you change anything on a modified release formulation, the agencies are going to require a BE study. If you have an IV-IVC built in a good one, you can get around a lot of those requirements. So I've mentioned this before. So the big thing is one of the big things you need to understand for modified release is colonic absorption of your compounds. It's usually needed for your modified release dosage forms. You have kind of a 2 to 6 hour transit time, which is highly intra and intra subject dependent. The graph to the left is looking at some of the stomach, or right, my apologies, is looking at some of the GI factors for somebody who's healthy and somebody who's kind of suffering from some IBD that day. So you could see a big difference in transit time. I think you can kind of see any pH at any time in the stomach or in the GI tract, depending on how you're going. You could see some microbiological differences as well. So, this is something that you'll have to kind of consider, is that, we talk about two-stage dissolution, and it's very neat with SGF and FASF, but in real life, it tends to get kind of messier with a lot of variability. And the reason understanding this is important is most of your extended release duration dosage forms, unless you're going for very short duration, are going to have some release in the colon. And most delayed release dosage forms will kind of creep into there as well, or a lot of them will be specifically targeted to the colon, depending on how long you're going to delay here. And it is a formidable barrier to drug absorption. There's very low water content, there's very low surface area, so it's somewhat of a desert. And there's a lot of enzymes in that area, so you're going to have to worry about enzymatic degradation of your drug. And the way we think about this is one of the first things we have to understand is early assessment of that potential and the absorption modeling there to even understand if this is a good candidate for MR. And there's a number of ways to do that, but kind of the simplest way without much work is simply looking at the developability class of your drug. So this is an older paper published by AstraZeneca where they looked at the human regional absorption data for 42 compounds, and they plotted it basically as a function of its BCS class. And what they kind of observed is if you're in a BCS1, you're in a pretty good spot. You're in a pretty good spot. You generally have very high colonic absorption. There are some exceptions to the rule, which they've kind of believe is due to enzymatic degradation of those particular compounds in the colon. If you're in kind of the BCS3 region, or 3 or 4, you're probably in trouble. You're seeing generally kind of a significant loss in bioavailability here, and that's really due to the fact that you don't, for the BCS4s, you're probably not being solubilized, and even if you are being solubilized, you don't have great permeability, and it's not a great environment for absorption to begin with. So this is something there, and then a couple of years later, Butler from GSK supplemented this by pulling an additional 21 examples out of the literature. This one he plotted by DCS class instead of BCS class, but sort of kind of confirm this, where if you're in kind of the BCS1, in this case DCS2A, which is a dissolution limited absorption, you're generally going to have pretty high colonic absorption, so you have a good candidate, good candidate for MR. If you're kind of in your low permeability classes, you're probably going to be struggling because you're not going to have good absorption. And the 2Bs, I called it variable. As you can kind of see, the error bar on this one's, kind of spans good to bad with some pretty big error bars. So, you may be in an okay shape with a DCS 2B, but that's something you'll have to consider. And also a DCS 2B, you're most likely have solubility limitations, so you're going to be, looking at a solubility enhancement as well. And then the other part that AstraZeneca's been more recently publishing about is microbiological stability. So the colon has your highest concentration of microbiota in the GI tract, and what they've observed is they've seen good candidates by kind of the DCS, BCS class end up being poor candidates in vivo due to the fact that the drug's being degraded. And what they've reported is sort of 19 of their 51 molecules in this analysis were, incorporated into human fecal bacteria, have degraded, and they've hypothesized that this is due to results in the loss of bioavailability. But at the same time, this actually can be used to your advantage. Some of your colon-targeted formulations that have been sort of developed by others have actually used this to your advantage by developing coats that are degraded in the colon to help release the drug. And then, so you can kind of build this into a decision tree. And this is one that was published last year by AstraZeneca. So what they're looking at here is basically, you have a formulation candidate with a target release profile. They first look at your permeability. If you have high permeability, they look at your solubility. If you have high solubility, you look at stability. And if you're kind of yes to all three, you're a low risk. And as you kind of start moving over, permeability ends up being kind of your first pass. And then it sort of looks at this where, if you have bad permeability, bad solubility, you're probably going to, fail to develop an extended release formulation. If you're kind of a yes, no, you're probably in a moderate risk here. It may be a suitable candidate, but, you need to be kind of careful here with your risk factors and address them in your development. And this is, kind of confirming this. This is the majority, this was, again, Butler just looking at how the marketed MR products fall based on their DCS classes. So as you can see, the majority of them are class 1 with, some class 2As. And there are some examples of, very few examples of 2B, 3, and 4. And it's also interesting that they're all kind of relatively short duration formulations. So they're not really relying on the colon, or they're, some of the ones hiding in here are delayed release as well. So, this also holds for most of your delayed release as well, in this case, enteric coats. So we talked about the API, and now what is your right technology here? So there's a lot going on here, but assuming you have a good candidate, you have kind of three general classes. You have your matrix tablet, which I'll talk about in a minute. You have your multiparticulates, and then your osmotics. So first thing is kind of just looking at your solubility. As you can see here, generally you need, for most of these, you need kind of, good solubility. The osmotics can somewhat handle the poor solubility, and this is, again, assuming you're not using a solubilization. Next part is kind of your dose strength. If you're in kind of a high dose, you're probably looking at a matrix tablet. Both the osmotics and the osmotics certainly have some variability. The osmotics generally cannot handle high doses. You just run out of room with those particular tablets. The pellets, if you're looking at kind of a conventional barrier coat, there have been more recent examples where you start getting to higher dose strengths using like very high drug-loaded, essentially mini tabs. But generally with your pellets, you're also kind of in a poor spot. And then you kind of have to look at what I would say is your performance characterization. Considerations and also what I'm going to call commercial and manufacturing considerations. Generally, the matrix tablets, you're going to be running into some bioperformance risks around food effect, around some of the robustness there. While the osmotics perform beautifully, however, the matrix tablets are generally easier to manufacture. You can generally use a standard process drain. The osmotics require basically very specialized commercial equipment, very specialized excipients, so they're much harder to do. And then the other part here is in the multi-particulates are kind of in the middle. And then your release duration, generally for a matrix tablet, you're looking at less than 8 hours, sometimes a little longer, but you have first order. Pellets, you can kind of mix and match much more to get what you want. And then the osmotics generally work better for longer duration. And with their particular mechanism, you're going to see both zero order release and also kind of a distinctive lag time. So if you need kind of a rapid onset plus extended release, a matrix or osmotic might not be your best choice. And then there are some, they're not commercializable, but there are a couple of options out there for diagnostic tools, which aren't commercializable, but you can run kind of a quick preclinical study or even a clinical study with these to run this. And what these are nice is you can remotely control the dosage form, so you know exactly when it's releasing and how much is left. And just a little bit on the on the hydrophilic matrix tablets. So these work by the drug is embedded within a polymeric matrix, which controls your drug release, and this will govern the drug release will be governed by your polymer and the geometry of your final dosage form. As I mentioned before, this works best for soluble APIs and can support a pretty wide range of doses, but they generally have bioperformance risks. And then from a processing perspective, most of these use standard tableting process trains. It's really just adding a polymeric excipient into your form, into your blending step. So, from a manufacturability perspective, it is more complex because you'll have to deal with the polymerics, but, you don't have specialized equipment or, kind of crazy parameters to deal with. The osmotics, these are considerably more difficult. In this case, your tablet is encased in a semi-permeable membrane with a laser-drilled orifice. So here's one up here. And then the release is driven by the hydration. So this is a push-pull osmotic. You have a push layer below, you have your drug layer. It's all encased in a semi-permeable membrane. In this case, generally cellulose acetate. You have kind of a laser-drilled hole at the top. Water influx in, your push layer begins to dissolve and swell and kind of pushes the drug out. So, here's kind of a typical release profile where you have a lag during that initial hydration phase, then kind of a more gradual release. And this is kind of what the tablet looks like as a function of time. The red is your push layer. The nice thing about this is the drug release profile is insensitive to a lot of things because it's all based on water ingress. However, there's dose limitations on the technology, and this is a very challenging manufacturing process. You need organic coating, you need laser drilling, and then you have kind of a limited excipient selection for your push layer in that semi-permeable membrane. And then the multi-particulate, this field really offers you a lot of versatility. So this is something our colleagues at Lonza put out last year, just the various triggers you can use and how you can design them. But basically your drug is in sort of a small pellet or matrix by a variety of different technologies. You can use drug layering, extrusion, spheronization. You can use mini tableting, and then basically the release rate's going to be controlled by core or the coating, depending on how they were built. And then they kind of get mixed and matched into a capsule usually. So it gives you really tunable release rates, and your gastric emptying time can be a little less variable compared to some of the big tablets. but your processing is a bit more complex. And then, so I've talked about this, and I've also sort of mentioned that you generally need a DCS1 or 2A or to do this. But what if you're kind of stuck there? So if you have kind of a 2A or 2B, your play here is basically solubilization enhancement. So you'd have to kind of develop your soluble enhancement technology, put this into the, your dosage form as well. And it does kind of add both technical complexity and bioperformance risks, but it's been done before. So this is, some examples from the literature of commercialized products. And what you kind of see here is you see some solid dispersions, you see some acidification strategies, you see some surfactants. So, this can be done. If you have low permeability, it gets very tricky, very tricky very quickly, to the point where, like, at least at Merck, if you come with a DCS 3 or 4 and ask us to develop it, we're basically going to tell you no. It's just not the right compound. But basically, your best strategy is to ensure absorption in the upper GI. That means you're going to have to cut your duration down. There have been examples of gastroretentives where the dosage, you make a very large dosage form that's ideally kind of staying in the stomach, and then really, drug diffuses out there, so you're just kind of keeping the upper GI saturated, for lack of a better term, a drug. So this is kind of a cartoon example here, and then you have sort of this is something Lindra puts out where it basically expands into a star, and then you can't, that will stay in the upper GI. There have been a number of strategies here, and I have some here. But what I would say is floating tablets or expandable dosage forms are the most common. It's questionable how well these actually work. Several commercial products have been reported as gastroretentive. They just tend to be very big, and then kind of have some limitations as well, which sort of questions whether or not true gastroretentive is actually being achieved, or it's sort of a very slow-moving tablet because it's big. And then just to pivot a little bit to your delayed release dosage forms, these are coated tablets or generally capsules that take advantage of your unique environment. Usually your formulation can use one or more. So your core tablet is kind of generally a standard IR tablet with a functional coat, and you can apply multiple coats to enable kind of multiple ones. This is kind of an example of a tablet that has an IR core, an erodible outer layer, and then kind of an enteric coat on top of it. And UJUKIT publishes a number of formulations. These are all generally functional coating, either pan or Worcester, depending on sort of your core particle or your polymer coating. And then, your unique challenges here is really you need very narrow and precise processing ranges. You often need organic solvents, which can, not all facilities can handle those at scale. And some of your film coats are a little tougher to compare than your typical kind of, non-functional coats. There are a number of delayed release capsules on the market. These are kind of two examples, but there's a number of enteric capsules on the market as well. In this case, this one is a Pulsin cap, which is targeted for the colon. You have kind of a water insoluble coat, and then it kind of pops off. The capsule top comes off, and then you have kind of a plug that ensures a little bit of delayed. And this is a guard cap from Avonic as well that has sort of a double-coated capsule, which has a pH-dependent and then a time-dependent layer. So kind of some of the takeaways I hope you guys take from today's talk is you do MR does offer significant benefit to patients, but comes at a kind of a cost of increased development complexity. Your first step is making sure you have the right compound or a good compound. BCS, DCS is a great first pass, and then you kind of have to think about your colonic environment. And then from there, you can kind of fit your compound to the best technology. And, I don't think there's one, solution that fits all here. Each formulation option has its advantages and drawbacks. I'd like to thank a couple of my colleagues, Philippos, Grace, and Tycho, and you all for listening, and I'll be happy to take any questions if I have time. I think I might have run a little long.
There's a question here. So first come, first served.
Okay, thank you. Hi, Joseph. Very nice presentation. Thank you for sharing this information with us. My name is Anand Ube. I work for AbbVie in Irvine, California. I have one question for you. Could you please comment on the propensity of API form conversion, crystalline versus amorphous, in some of the platforms you went over, or is there any preference for?
So in terms of API form conversion, I would say it's probably the same as any general dosage form. It's something you'll have to keep an eye on. It's something you'll have to keep an eye on. My experience has been generally for our crystalline drugs, if we're picking the right polymorph to begin with, we're not seeing any sort of problem. Beyond that, if you have a drug where you have significant, let's say, crystalline, instability or polymorphism, that, will often just be as a challenge as well. Some of these technologies, you are kind of exposing the drug to, especially if you're like melting it or something like that, that is a risk that you'll have to kind of look at and, look at the API properties. But I don't, generally physical crystal form's not like a unique consideration.
Thank you.
All right, thanks very much, Joe.
Thank you, Joe. So it's my great pleasure to